Welcome to the webinar on Epstein-Barr Virus, or EBV, a current hot topic in MS. EBV is being considered a likely cause of MS for many years, and that's a view that's recently been strongly supported by an impressive study published earlier this year that received a great deal of publicity. In addressing this issue, we're very fortunate in having two international experts who will cover different aspects of the topic, who is Professor Mitchell Wallin from Washington, D.C., Director of the MS Centre at the Baltimore Veterans Administration, that's VA Centre, and also Associate Professor of Neurology at the George Washington University School of Medicine. Mitchell has undertaken extensive research in MS over many years and also in nervous system infections. So he's ideally placed to give an overview of the relevance of EBV infections in MS. Mitchell, I hand to you.
Thank you, Ernie and Amanda and the meeting organizers here. I'll try to key up my slides here. I'm going to give a little bit of the clinical and epidemiological background for why Epstein-Barr may be in the causal pathway for MS. I have no disclosures to declare, and this is the three things I'd like to hit with my brief talk. Review some of the key risk factors and clinical features of MS and how we can arrive at a viral cause for MS and what the underlying hypotheses are based on the clinical and epidemiological evidence, and then talk a little bit about Epstein-Barr and its association with MS. And basically, I can focus on the study that Ernie referenced from Ascherio Group at the Chan School of Public Health at Harvard, and kind of wrap up. Many of you know the MS International Federation. It's a worldwide group interested in promoting MS statistics and needs. There was an update done a couple of years ago, 2020. I was part of this group, and the new estimate is there's 2.8 million people around the world with MS. So that's 1 in 3,000, about 0.3%. When we looked at many areas of the world, about 3/4 face issues related to diagnosis, and really many parts of the world, there's not the neurological expertise or MRI imaging available to actually make a diagnosis, but that same number have issues with accessing treatment and symptomatic care as well. So despite the fact that MS is more recognized, there's many places where people can't get diagnosed and can't get access to drugs. So just point that out. If we look at the risk factor picture on the right side here, there's a number of environmental factors that have been associated with MS, Epstein-Barr being one of them, but all these factors have been shown in various studies to modify the risk for MS. And on that backdrop, you have the underlying genetics, gender being a major issue with female preponderance in MS. And then we have disease modifier and susceptibility genes. We're finding mainly in the HLA locus where immune genes are based. But there's been over 200 single nucleotide polymorphisms, small little gene SNPs that have been associated with MS that have fairly small effects, but together they also contribute to the genetic risk. If you look at recurrence risk for relatives of MS, this is sometimes the most dramatic changes you'll see in MS. We talked about the general population risk of about 0.3%, but you go up by about 10-fold when you get to a first-degree relative, around 3%, child or parent, and then another 3-fold increase to a twin, a dizygotic twin, and then up to 30% for a monozygotic twin. So this is some of the most dramatic changes in terms of sharing DNA that we've seen, and this has been put together in a number of different studies, but this is kind of the summary. So sharing genes can matter for MS, and but it's at maximal about 30% of the equation. The biggest risk locus genetically is the HLA locus, especially the DR area. And here we look at the odds of MS, and that's this middle one here, with the 1 being unity. And if you carry the 08, 16, or 15 allele, you have up to a 3-fold higher risk of getting MS. Some alleles in the HLA locus are protective, and it's fairly complex. And I think a lot of investigators are looking at some of these subtle changes in the HLA alleles that may drive MS risk or protect somebody from MS risk. That's the backdrop of genes, very in a basic way. But what are the environmental determinants for MS that give rise to looking for a putative cause? So there's been geographic gradients that have been described in North America, Europe, and Australia. Also, MS incidence and prevalence. Incidence is new cases of MS. Prevalence is all are fairly low in the equatorial zones or tropical zones. So where you live matters. And also, we know that migration, when you move from one place to another between regions, that can alter your risk for MS, indicating that environment is important when you move, and the time you move is important if you move before or after adolescence. And finally, MS epidemics have been described, and I'll just highlight a couple of these here. So here we have prevalence gradients. This is from the same MS International Federation journal that looks at the world in 2020. The red areas are either above 100 or above 200, and North America, Western Europe, Australia, and New Zealand are all high MS prevalence zones. The gray areas are where the group actually found very limited data, if any data. So sub-Saharan Africa, Greenland, and also parts of Asia really have poor surveillance, or at least unavailable surveillance over the last few years. And this B side here just talks about the confidence in the numbers. And so those same regions where we don't have data are the same regions where we need to actually go and try to find better surveillance and reporting for MS. But where you live matters. This is just one study. There's been several that have been described that have been well put together. This is a migration from UK to Australia. This was just put out a few years ago by Barnett and colleagues. So the Australia-born MS cases were assessed basically in Hobart, Tasmania. And if you look here at the graph here, they had a period from 1941 to 1965 as early onset cases, and then 66 to 1981. And so what they found was a doubling of the incidence rate in immigrants that went and traveled to Australia in these early years compared to this basically next generation. And most of the immigrants migrated after the age of 15. And for those that did, the interval between immigration and year of onset was about 6 years, versus a much larger period of time, 22 years for those migrants that migrated under the age of 15. So adolescent period seems to matter with your risk for developing MS. So basically the people that have migrated after 15 got caught up in the environment of UK and brought that risk with them. And you can see here, this is just the prevalence data looking at the specific prevalence rate per 100,000 in Australian-born in Hobart. And you see the dotted line here is the later onset group, and the solid line here is the early onset group with a lower prevalence. So this study supported a transmissible factor, environmental factor, associated with the rather dramatic increase in rates in this very well-studied region of Tasmania. Probably the best studied epidemic was the Faroe Islands epidemic, and this occurred in a small group of islands. You can see it highlighted up here between Iceland and Norway. And these are part of the under the Danish government, and that's where they get the medical care and the statistics. But John Kurtzky here in the left here, we have Ann Helperg and Kay Hillstead were the three that really put a lot of the data together. And they went back and looked at all the cases from early 1900 to basically going forward and described rather in a number of papers, four-tiered epidemic. The first big curve occurring during World War II. And what the hypothesis was is that this epidemic was actually associated with British troops that were stationed on the islands during World War II. And they actually proposed a widespread but persistent infection that was driving this epidemic. They called it PMSA, or the primary MS infection. But probably the best described epidemic of MS in a classic island location where there's little in and out migration. So when we look at hypotheses for MS, when it comes to the environment, two main types drive it. So prevalence hypothesis espoused by John Kurtzke, that MS is the result of an infection more common in regions of high MS risk. We showed those areas of the world just a few slides ago. And then alternatively, there's the hygiene hypothesis that was first put forth by post-cancer and later others. MS is the result of a common infection acquired at a later date, resulting in increased risk. So both of these have some support, but we'll kind of move our way forward. So why a virus? We talked a little bit about the epidemiologic evidence and the fact that the identical twin concordance was only 30%. There are a number of animal models. The most well-studied is the Theiler's virus, the murine encephalomyelitis virus. And also there's no genetic disorders that we know of in which the brain and the CSF compartment produce oligoclonal bands. So Epstein-Barr is a herpes virus. It was discovered in 1964 by Dr. Burkett, who was stationed in Uganda, and lymphoma was named after him. It's acquired through the saliva, and it can actually, it goes into infecting B cells here, and which can multiply and then interact with T cells, and it undergoes a latent stage and can be persistently shedding virus in the mucosal areas or in other parts of the body. But it's been associated with a number of cancers: gastric cancer, Hodgkin's lymphoma, nasopharyngeal carcinoma. These have all been well studied and honed in with very good epi and clinical studies that made these associations over many years. And recently, too, it's been associated with lupus, which is a similar disease to MS in terms of type of acquisition, demographics. So the adolescent manifestation or young adult manifestation of EBV is mononucleosis. And there's several similarities to the clinical epi of MS. It occurs maybe slightly earlier in terms of age of onset than MS does. The sexes has a female preponderance. It has a lower incidence in non-whites. There is a latitude gradient for both diseases. And it's also associated with high SES. This slide shows a meta-analysis of mononucleosis, acquiring the infection as an adolescent or young adult, and risk for MS. And overall, these studies, you have a doubling. This means the relative risk here is basically around 2 times the risk if you have a history of mononucleosis versus if you don't for developing MS. So this was one of the earlier studies that actually put together some of the literature showing the association, and it's been pretty consistent across most studies. This is a nice review done by Ben Jacobs in London, looking at EBV and its associations. He looked at every study where there was data available in children or adults for EBV serology and looked at risk for MS. And again, this plot here, this is middle line here is no increased risk, and here we show about a 4-fold increased risk for MS if you have EBV serology that's positive. And moving on to the study that really got a lot of attention earlier this year that Oni talked about was basically a compilation of data from Ascherio Group and to show the association of EBV with MS. And what he did is he collected data from the US military with active duty military personnel. And these are some of the demographics. As opposed to the MS population, the military population has a male bias. So it's basically about, depending on when you survey, it's about 20% right now, female versus and 80% male. So that's by nature what this is the background active duty population. This is the age range. This is the race and ethnicity. And the race and ethnicity of the military really mirrors the US population by and large. And the study population that he was able to gather from the military population is depicted here. And so he got both clinical information and also serum. And the powerful thing is that the serum, if you look down here, this is the age at first sample, was 10 to 20 years prior to the age where MS was developed here. So this is the green, the age at first sample, blue is the age at MS onset. So the beauty of this repository is that it can actually show blood and any kind of markers that would be developing prior to the onset of the disease. And the category C here is the time to MS onset and the first, second, and third sample. So they were able to get up to 3 samples on every MS case, along with a matched control prior to the onset of MS. And they did a series of experiments to show that EBV is associated with MS, rather elegant experiments. That's the background. And this is a very powerful resource because the military has right now 75 million samples right now that are being collected. And really started right after the HIV epidemic happened in the 1980s. Every military recruit in active duty gets a sample at least once a year, or in some cases every other year, and it goes to the storage facility after they test for HIV. So this is EBV infection showing how it precedes MS onset. So they were able to do EBV serology on each of these serum samples. And if you look at the first, second, and third sample, the green here is cases and the blue is controls. You can see there's a separation at the second sample with EBV titers. They looked at CMV to look at a confounding virus that is spread in a similar way to MS, and they didn't find any significant change here in the 3 samples, all obtained prior to MS onset. And the lower graphs here just show the risk here of EBV seroconversion for MS. And you can see when they seroconvert here, the risk goes up 20-fold compared to baseline. And this is the absence of risk here shown with the CMV titers. The next is to show how neuropathology could be developing in relationship to the EBV infection. So the graph here on top A looks at NFL. This is a biomarker for central nervous system damage or pathology. It's been used commonly in traumatic brain injury, but increasingly with MS as a biomarker for pathology. And you see basically the time points here before EBV infection in the first bar here. This is at the time of first EBV positive sample, and then NFL after EBV infection. You see this dramatic rise here that occurs after the EBV infection with time, And then the same is here for controls here on the right. So with MS, you see this dramatic increase in the titers and then no change here with controls. So rather elegant way to show early, at the earliest possible stage, even prior to MS onset, you have potentially neuropathology ongoing in relationship to the EBV seroconversion. And finally, to show that other viruses are not are involved or not involved, there was a fairly sophisticated virome scan that was done in 30 cases and 30 controls, pre-onset samples. And so what you see here is the pre-onset samples here are on the left, post-onset samples on the right. Actually, the titers here and the type of viruses, this is actually a very sophisticated viral scan where they can actually put serum or other bodily fluids into wells, and they have most every virus that is known to cause pathology. It's a rather extensive library. And essentially, they came up with this map here, looking at pre-onset and post-onset. So Epstein-Barr virus had the most hits in terms of number of peptides, but there's a number of other viruses that we see here too. Other human herpes viruses, 1 and 2. There's some adenoviruses and dengue viruses, a number of other, but by and large, the peptides from EBV were present in both the pre and the post-onset sample. If you go to the bottom here, you'll see EBV antigens. The biggest one is the EBNA1, which is implicated in pathology, both in the pre and the post-onset samples, was the most well represented, but other EBNA peptides were there, as were a few other capsid peptides. Brian Jacobs and the group from University College London looked at a few interactions in his meta-analysis of EBV. On the left here, you see the relationship between infectious mononucleosis and if the HLA locus, DR locus was positive. Essentially, the one here is the increased risk threshold. So if you were IM negative, infectious mononegative, and HLA negative, you did not increase your risk. You had a little bit of a bump here just over unity with infectious mononucleosis without the HLA factor. And then with the HLA alone, even positive, increased risk maybe up to 3-fold, but you had an additive risk with both positive. The same thing happened with smoking, showing the additive risk here, not multiplicative, but there is basically there's showing here that the relationships are additive to other risk factors going along with EBV and HLA in the models. I'm not going to get a lot into mechanisms here, but this was a recent paper that was put out by the UCSF group last year. And there's a series of HLA interactions here. And there's panel A on the top there shows that multiple sclerosis associate HLA, the DR15, the class 2 locus, and how it actually sits on the side of a B cell. So, and then on letter B here, you see that the role of these HLA molecules in interacting with the DRB2 antigens in T cells. And C shows, the level C here shows, excuse me, my mouse keeps jumping here, but letter C shows how these T cells can interact both with the B cells and also other antigens to create a self-peptide memory. And sometimes there can be higher avidity or foreign peptides that can be involved in this mix to activate P cells to self-antigens. So this is the potential mechanism how you can have involvement of the HLA, potentially EBV particles that may mimic or provide antigen discovery for T cells to go and attack other human cell proteins. So in sum, these results support a causal role for EBV to increase MS risk. Higher risk is associated with later age of acquisition of MS. Potentially, an EBV vaccine could prevent many MS cases. And right now, the Moderna company is in a phase 1 trial with an RNA vaccine. Also, EBV-directed therapy could potentially alter the course of MS. And my colleague, Dr. Smith, will talk more about that. Some things that aren't totally explained by the EBV hypothesis, the growing incidence of MS in non-white populations. Because EBV is not as common there, you have to have another hypothesis. Are these individuals getting infected in a different way by a different strain? The Faroe Islands epidemic by nature is not a late acquired infection, but a common infection that's spread. And also the changes that we see, just like we had that study from UK migrants to Australia, you have to implicate some other kind of more widespread infection. That's something that comes on later in life. This is just some military data that Deucine and our group did, showing the incidence rates of MS in Gulf War veterans. These are people that served between 1990 and 2007. And so here we show really for the first time that Blacks have a higher incidence rate than whites, and Hispanics and other races actually come fairly close to that too. The same thing was shown over here. Blacks have the highest rates here in this active duty segment here by Deucine. So it's hard to explain that. It's just been what appears to be a relatively recent change over the last 30, 40 years. But you have to implicate this to explain how a virus can be causing this in populations where they have traditionally acquired early in life. Other viral candidates that have been put forth in addition to the Epstein-Barr virus include human herpes virus 6, A and B. And this has been gone through a number of elegant experiments that show it can produce neuroinflammation, demyelination, impaired remyelination, and may actually trigger EBV. On the left here, you see human-derived astrocytes with the little stars there showing HHV6 surface, the little proteins here and particles, and then the surface proteins here are the GP116 there. The blue or the green is a surface marker here. The blue is the astrocyte marker. So these have been seen and tracked in cells. They've been found in brains of patients with MS. And there's been a number of fairly elegant experiments, not to the extent that EBV has undergone. We're still kind of coming through this crazy epidemic with SARS-CoV-2, but there's been a number of reports implicating either the vaccine or the virus itself in first onset of MS or NMO or other demyelinating disorders. So people are actively investigating how SARS-CoV-2 through neural infection, olfactory, its tropism and getting in the brain, and through its lymphocyte imbalance and colonization of the GI tract, how it may be influencing MS and or the EBV. It also has been associated with Parkinson's and narcolepsy. So our group, I've been working with the UCSF group for a while, and we will present some data next month on our own cohort. We have a similar cohort of military veterans with pre-onset MS serum, and we are trying to just duplicate some of the things that have been done with Alberto's group. But this is the first data presentation. Those are going to ECTRIMS next month. So with that, I think I'm about done with my time here. We can take some questions at the end. So thank you for your attention. I know I can't see everybody, but thank you for listening.
Many thanks, Mitchell. That's a very clear exposition of a complex topic. Certainly interesting. Our second speaker is addressing more specific issues relating to EBV and how it might be managed. Professor Corey Smith is from Brisbane. He completed his PhD in microbiology and immunology at the University of Melbourne, and for a number of years now has been based at the Queensland Berghofer Medical Research Institute, where he heads the Translational and Human Immunology Laboratory, where they're focusing on translating basic research to the clinic, a very important aspect. Corey's particular interest has been in T-cell mediated control of human viral infections, including EBV. This is relevant for a number of cancers as well as MS. He's been involved in Brisbane with some innovative clinical trials and I think the potential commercial development of T-cell therapy targeting EBV and MS. And I'm certainly interested to hear an update of that work. Corey.
Thank you, Ernie. It really is a pleasure to be able to talk to you all today about the work we've been doing in developing therapies for EBV. I'll just get my share slides sorted. Here, as I mentioned, it really is a privilege to talk about some of the work we've been doing. So just to give a brief background, so we're So I'm at the QI Burkhoff Medical Research Institute in Brisbane, Australia. And I've been part of it's actually a really long-standing group that's really primarily focused on viral immunology. So understanding how our viruses interact with our immune system. And, it's really been 4 to 5 decades of work from, some of my mentors looking at particularly Epstein-Barr virus and how it interacts with the immune system. And this has led to our work, particularly initially in collaboration with Professor Michael Pender, who was a neurologist at the Royal Brisbane and Women's Hospital, looking at trying to target EBV to treat multiple sclerosis. So today I thought I'd just talk about give you a little bit of background on EBV and how, maybe it's contributing to MS, but also, the therapy we've been developing, and finish off on potentially on some ideas around probably the other approaches that people are thinking about for targeting MS. So some of the work today I'll talk about is work we've actually been doing with Atara Biotherapeutics, which is a biotech company based in California in the US. And I was, very lucky to get involved fairly early with this work. And, that they quickly saw an interest in potentially, looking at translating some of our research on EBV T-cell immunity into treating patients with MS. And so I just have some of those disclosures. So as Mitch went over, EBV is as you mentioned, was discovered by Burkitt and Epstein in the 60s, but it's actually, as many of you will be aware, it's usually a childhood infection. So in most parts of the world, we acquire the virus in early childhood. But as was mentioned, really, the risks of symptomatic disease arise really in adolescence and early adulthood, where about half of those infected at that stage actually go on to develop what we would traditionally call glandular fever or infectious mononucleosis. But actually about 95% of individuals are probably around the world are infected by the age of 40. And this leads to a lifelong, persistent, stable infection in our bodies. But what we do know from a long period of research is it's actually the T cells in our body. So these are the what we traditionally would call the cytotoxic T cells or the killer T cells that play the critical role in the immune surveillance of these EBV infected cells, these latently infected EBV cells. So we've actually done a lot of work over a long period of time understanding how EBV interacts with our immune system. So these are 2 graphs are really just kind of representing the type of immune responses we generate after we're exposed to the virus. So essentially and this is really represented best in those settings of IM or glandular fever, where obviously these gray bars represent that period when we might have glandular fever. And what this is actually primarily driven by, it's not just driven by the virus, it's actually driven by this huge expansion of a certain type of T cell and with certain reactivity to the virus within our bloodstream. And that's why it's actually called mononucleosis. It's the dramatic expansion of these T cells. But these T cells actually recognize the life cycle of the virus that's involved in generating lots of virus particles. And these are what we call lytic antigen T cells. So these, because we've got lots of virus in our body, these then, float around and generate huge populations of these T cells that we actually don't necessarily think play a huge role in controlling that persistent viral infection that we all end up with. And what plays the most important role at that stage is what we call these latent antigen specific T cells. And I'll come back to this in a little second when I kind of explain what EBV does in our bodies. But generally, these are a little bit slower for us to make, and they generally tend to come up a little bit later. But what is important to note is when we maintain EBV in our body for our whole lifetime, we maintain these T cells as well. And it varies in levels, and how that associates with risk of multiple sclerosis and other diseases is really still something we have to understand a lot better. But what we also know, something we actually did again a few years ago, is the other interesting arm of our immune response to viruses is what is our antibody response. So this is, many of you would have obviously heard about SARS-CoV-2 and the vaccines and the way they're supposed to work. They're really about driving what we call these neutralizing antibodies, which prevent the virus from infecting more cells and prevent it from being spread. actually, we noticed, and it was actually, noticed previously a long time ago, that we actually don't do this very well with EBV. We actually don't make good antibodies that neutralize the virus. And you get, these only really come up late at these later stages. You do make other antibodies which are used to diagnose the infection, which is against just the other parts of the virus. And also, as many of you may be aware, we also start to make antibodies against this EBV nuclear antigen 1, this EBNA1. And these are the ones which are associated with potentially with, multiple sclerosis risk. But again, we don't think these actually play a role in controlling the infection. They're more likely a consequence of us having these virally infected cells in our body. And whether it's an indication of how many virally infected cells in our body, that still needs to be completely elucidated. So EBV is actually associated with a lot of diseases. Really, initially it was associated with Burkitt's lymphoma, which is where it was identified. And these are diseases that arise in our B cells, which are the cells that make antibodies. And I'll come back to how EBV does this in a second. But a lot of these are cancers, okay? And these cancers arise in B cells from the uncontrolled proliferation of these cells. And, one of the, another important one is these cancers that arise in patients who've had an organ transplant. So they have a suppressed immune system and they end up with these lymphoproliferative diseases. But in actual fact, it also causes other cancers, which are kind of in atypical cell types for EBV. And these are ones we've worked on for a long time that are actually prevalent in Southeast Asia. But also it's associated with other diseases. And, multiple sclerosis probably fits in this immunopathological diseases with, infectious mononucleosis. It also causes some diseases actually associated with some genetic disorders, these excellent lymphoproliferative diseases. So EBV really is associated, with a lot of disease around the world, and there's probably more, but it hasn't been easy to link the EBV to diseases like multiple sclerosis because of its prevalence everywhere. But I think, as Mitch pointed out, these are starting to become clearer. So I thought, I wanted to try to give you an idea of what EBV does in our body and how we think we can target it by trying to target using our own immune system, or in later settings, the immune cells from other people to try and treat these EBV diseases. So as I mentioned, EBV actually infects the B cells in our body. And these are the cells in our body that normally make antibody. So what happens normally when a B cell changes to become an antibody producing cell, it actually matures through these different phases. It doesn't really matter too much about the specifics of this, but they then go on to become these plasma cells. So these plasma cells are the ones that make antibodies that protect us against viral infections. They'll go and protect us against exposure to SARS-CoV-2, etc. But what EBV actually does, it actually co-opts this pathway for its own purposes. So it's actually, the virus itself is actually able to drive our B cells into these different pathways to allow it to survive within our bodies. And these are linked very specifically to what we call these latency profiles, okay? So these are just different profiles of how the EBV functions in these cells. And eventually, as you get to these later stages, the EBV infected cell may mature into a plasma cell. And we think these are the ones that then start to make more virus. And this is the virus that gets shed through our saliva, etc., and spread to other people. But what is also interesting is that actually these type of latency and these infection types within these latency profiles within these different B cells are actually associated with different diseases. So, so the most, as we focus on is these B cell diseases that we know about. So for example, what we call this latency 3 type disease is actually present in patients who have post-transplant lymphoma. And these are cells that really can proliferate a lot. They grow a lot in our bodies. Because we have no immune control. So essentially, if someone has a transplant, you suppress their immune system, these cells grow uncontrollably. But I should point out, one thing that's become clear is that you can actually use T cells from other people, particularly, to actually restore control of these EBV proliferating B cells. And the other types are obviously the things like Hodgkin's lymphoma, which has shown some evidence of responding to therapies, and Burkitt's lymphoma, which is actually more difficult to target because it really doesn't have many EBV proteins, just that EBNA1. So that EBNA1 protein, which is, kind of a biomarker of antibodies and risk of MS, is actually critical for the virus. The virus has to have this protein expressed for it to divide between the different cell types. So we actually think, based on some of the studies done looking at EBV in autopsies in patients with MS and other settings, we think potentially the type of EBV infected cells in multiple sclerosis are more likely to have this latency 2 profile, where we express these proteins of EBV, rather than being really fully immunogenic with all these other proteins that our immune systems actually see really quite well. This is really generally still a hypothesis, but it's really the hypothesis that underpins our approach to seeing whether we can use T cells to target patients, to augment and improve, the MS patient's immune system, and to remove these EBV infected B cells to see if it'll have an impact on their disease. And so obviously the idea is if you can eliminate these cell types, can you have an impact on disease progression? But obviously, I think ultimately the goal is to look at strategies, as Mitch mentioned, potentially of vaccination, which may prevent or reduce the burden of EBV upon infection. So what I want to talk to you today about is our studies looking at what we call adoptive EBV specific T cell therapy. And this is essentially where we generate T cells in the tissue culture, and then we reinfuse these into patients and obviously have a look at the outcome on their disease progression. But particularly early is really obviously all about safety. So this is essentially the way of how we started with our adoptive T cell therapy. And this is, there are really 2 ways you can do these sort of T cell therapies. Is this autologous approach, where you're taking material from the patient themselves, or as I'll explain later, is this allogeneic approach, which we're using with Atara, which allows you to store large quantities of cells from healthy people that you can then administer to patients as we move forward. But essentially what we do is, so in these initial studies we did, we recruited MS patients. We then use their peripheral blood and we isolate their white blood cells from this blood. And we use a strategy we have that allows us to expand these killer T cells, and we can then infuse these back into the MS patient. So the first patient we treated with MS was actually recruited in 2013, and this was a study that we published in 2014. And this was really driven by Michael Pender, who really has had this strong hypothesis that the reason EBV causes, it was associated with multiple sclerosis, is that the patients have these EBV infected B cells in their brain. So this was actually a patient that had his first MS attack in the early 90s. He'd then gone on to have secondary progressive MS and had a very high EDSS score. So essentially my role in this is to developing the platform we use to make EBV specific T cells. And the cells we generate, recognize these EBV proteins, and then we use these T cells as a treatment in this patient. And what was really promising was the potential association with improvement in his symptoms in this first treatment. But obviously what we're also very worried about is putting immune cells into an environment of essentially immune pathology and the risk that might cause. But we didn't see any adverse effects of our treatment. What was particularly encouraging and what drove us into our next early phase 1 study and what's driven Atara is some of these really interesting results that occurred in this patient. And whether these are coincidence or associated with our treatment, this remains to be resolved as we move into phase 2 and hopefully as Atara move into phase 3 studies. But what's interesting, we saw this really reduction in these brain lesions, but also the reduction in this the quantities of IgG in this patient. So this was really interesting. Suggested to us, there is potential that by using EBV T cells, you may be able to target those EBV infected B cells. So we then, from that, we then moved on to a proper phase 1 trial. I have to say, this was supported by MS Australia and MS Queensland, which we're really grateful for. And this trial was designed to, again, as a safety trial, to show that if we use these T cells, we aren't going to cause any severe adverse events associated with, potentially exacerbating disease or other potential side effects. So this was a trial with 10 MS patients. 5 of these patients had secondary progressive MS and 5 had primary progressive MS. So this again is in that autologous setting where we grew T cells from these directly from the patients, and then we reinfused these into our patient cohort. And again, what was good, this therapy was really well tolerated. There really weren't any reports of adverse events. And of the 10 patients we treated, 6 experienced symptomatic and objective neurological improvement, again, in an open label, uncontrolled trial. And, a 7th patient actually showed some improvement. But what was interesting is that it seemed to be associated with the strong reactivity in the cell therapy products we make. So the way we generate these cells and the potency or the reactivity that comes out at the end is really dependent on inherent in the material we kind of start with. So obviously in a few individuals, we can only generate what we would consider a weak T cell response. But I think what was particularly interesting in this is actually represented in this figure in the lower right-hand side. This was the initial patient we treated originally in 2013, who actually was again enrolled in our study and received a second round of treatments. So this is just looking at their IgG concentration in the CSF. And as we mentioned, what you can see, so these little lines across here are actually the injections of the different T cells. And you can see under that first treatment, you can see a reduction in that in the IgG in the CSF. And then you can see it kind of plateaus over an extended period of time, and then it started to increase. What was really fascinating after the second round of infusions, again, we saw this dramatic drop in the IgG in the CSF for these patients. And obviously, we also saw some improvement as well as in other symptomatic improvement. So again, this suggested to us these therapies are safe and have some potential for MS patients, which was really fantastic. And it sort of starts to provide some support for Michael's hypothesis that it is these EBV infected B cells that are still contributing to the disease. So where we're at now is that around this stage, we'd already had some discussions with Atara Biotherapeutics, as I mentioned, a company in California, about some of our other work, as well as our MS work for EBV therapies. And their approach is actually to use, generate T cells from healthy people. So the idea is that this is what we call an allogeneic EBV T cell therapy, where you take blood from healthy people. We use these to generate our EBV specific T cells. And then essentially like what we would do for organ tissue matching, that they're essentially, closely matched to the patient, so that when they're infused, you can have this strong tissue matching, which is actually critical and which actually obviously is linked to the HLA profile, which is critical to allow for that ability of the T cells through their T cell receptor to recognize the EBV antigens on the EBV infected B cells in the patient. And the reason we're keen on this approach, and I think Atara is very keen on this approach, is that you can have much better control over the quality of the cells you make and much more consistency. You can have some problems, obviously, in patient cohorts that have other complications, and maybe there's a natural immunosuppression of EBV immunity in MS patients, which has been suggested, but it's not completely clear. We can scale this up to large numbers, and obviously we can use these T cells then fairly quickly in large numbers of patients, and obviously potentially redose patients over an extended period without having to remake T cells. So this is just an update on outcome from their first phase 1 study. So this is again this is looking at the follow-up in these patients. So again, these are all progressive patients. So this is all data that Tara has published, and this is just looking at what we would call a swimmer's plot, looking at EDSS improvement over time. So what is really interesting in a cohort of these patients, if you look at this dark green, there is evidence of EDSS improvement. In 7 of these patients, and in some of these patients, this is still ongoing after 3 years, which is really fantastic for our phase 1 study. And if you see in this light green, the majority of the patients that were treated show some evidence of stable EDSS, with a small number continuing to, unfortunately, have progressive disease. So again, providing some interesting observations around whether we're potentially, have a strategy to target EBV in our patient cohort. So this is just another way of looking at this, and I quite like this figure. This is again a figure prepared by Tara. This is looking at those 7 patients and looking at their improvement in EDSS score. So what you can see is, and this is, in all of these patients, you see this improvement in EDSS score across time with some a reasonably significant improvement in some of these progressive patients, which again suggests to us that there really is some potential in this approach, and it's why we're continuing on with this study, and it's why Tara has now moved into a phase 2 study. So this is what they call the EMBOLD study. So this is currently, this is a study at 35 sites in the US, Canada, and Australia, and this is actually a double-blind study, which is it wouldn't normally or commonly occur in these type of T-cell therapies, where usually you're in a situation of treating cancer patients where they would be just enrolled and there'd be other approaches to looking at controlling. But essentially in this study, 40 patients have received what we call ATA188. That's actually the cell therapy's name. And there's actually a placebo arm of 40 controls, and they'll get assessed at a 12-month time period. And in order to offer treatment to those controls, as placebo controls, they will then cross over to 188 after the 12 months, as well as the initial 188 will be redosed, and this will be the open label extension. So these will then hopefully be, following up for up to 5-year period, really with looking at the primary endpoints of controlling EDSS, or whether stabilizing EDSS or seeing improvement in EDSS. So currently the aim is that hopefully that Atara will be releasing results on this study around October next year, I think, with the hope that they'll be planning to move into a phase 3 study after that, to see if we can see evidence of strong statistical evidence of control of disease. So this is all quite promising at the moment. We just have to wait and see how this plays out. So that's kind of given you an oversight of how we think we can target EBV with T-cell therapy to treat multiple sclerosis, and if that premise that it's these virally infected cells that are remaining and that can be eradicated is playing a role in the ongoing exacerbation of disease. There'll also be some work that's probably going to come up looking at antiviral drugs for EBV, and this hasn't been a particularly easy area of actually focus of EBV research. So these are the antiviral drugs that people may take for other common herpes virus infections, such as acyclovir, which you might take for cold sores, or ganciclovir, which is a drug that's often used for things like CMV when it arises in transplant patients. These viruses, these drugs haven't had a great success against EBV in settings of the post-transplant diseases, but again, it isn't. It's probably a different scenario in MS, and these drugs tend to target that lytic cycle of the virus. So if that is playing a role in disease exacerbation, then you can control that. Some of these drugs may show some potential benefit. And I think, as Mitch mentioned, the potential for vaccination is there again now, potentially for EBV. The application across, epidemiologically, I think is an interesting question about who you target and when you target is going to be very interesting to try and prevent the risks. But I think there's a focus a lot with Moderna and NIH and things on these preventative vaccines. There may be also some potential approaches on trying to design therapeutic vaccines that may do something, similar to what we hope ATA188 is doing. So again, the idea really is, for the EBV vaccines is, can we improve on what we do naturally? And this is, not generating this strong neutralizing antibody response, and actually having antibodies in our body before we potentially go on to have IM symptoms or, other primary infections with EBV. And, you know, whether we could ever remove EBV completely, it seems unlikely. But if this could reduce those, you know, symptoms, and if those primary symptoms of IM actually, and which the evidence suggests are associated with MS, that, you know, we may be able to reduce the burden. The other area which we're interested in still is, you know, trying to improve these latent antigens specific, you know, T cell responses, as I mentioned, and you know whether we actually also boost these, both prophylactic or therapeutically, can they then enhance, you know, prevent the risk of developing multiple sclerosis, or in the long run, you know, maybe, you know, therapeutically, like our HA188 may be doing, is control disease burden. So, as I mentioned, there have been some vaccine studies done in EBV. You know, many of these were completed quite a while ago, really trying to make neutralizing antibodies. And these actually showed some efficacy, particularly these later studies, these ones in 2007, at reducing the risk of glandular fever, which was really, really quite promising. Obviously, they didn't reduce the risk of infection, and, you know, they didn't progress any further. But this kind of approach is what's starting to come back now. And actually, our group in Queensland, before my time, actually did a study looking at a vaccine to try to target T cells. And again, there was some suggestion that you could reduce the risks of developing glandular fever. But really, at the moment, there's two main clinical studies. There's obviously Moderna's study, which they're entering phase 1 studies, but also the National Institutes of Health in the US are also, you know, looking at a phase 1 study. And again, the focus primarily on is neutralizing antibodies. We with Atara do have a strategy in preclinical development that we're hoping within the next few years, depending on the outcome of these other studies, that we may be able to look at, you know, moving into phase 1 trials. And what we think potentially the approach here, the benefit of the approach, is looking at trying to target those T cell responses. So I hope I've just given you a good general overview of how EBV interacts with our immune system, and, you know, the potential role it may be playing in MS. I think there's still a lot of work to do to understand the relationship between those EBV infected cells, our immune system, the immune response, you know, linkages to all those HLA associations, linkages to the effect of, you know, vitamin D on EBV specific immune responses. But I think, you know, what has come out lately is that this really strong relationship. But from my perspective, you know, I just want to finish off with some really key acknowledgements for us. I think we were really lucky early on when we started this work, and to have good support from MS Queensland and MS Australia, which is, you know, continues now. And we're really appreciative of that, particularly with this, which is probably a somewhat controversial idea, that you could apply cellular therapies, apply T cells into these patients. And also we you know, we're lucky enough to get a lot of support from different organizations within Australia that support our cellular therapy programs. You know, and this is really the group that has led this work, and contributed to this work. And really, that's the driving forces behind this were some of my mentors, Professor Rajiv Khanna and Scott Burrows, who did a lot of the EBV immunology work. Rajiv drives our cell therapy program, but also Michael Pender, who's the one who's really pushed this theory of EBV and MS and the relationship between these EBV infected B cells in the brain, and his colleagues at the University of Queensland, but also our collaborators at Atara. And I'll just, you know, finishing off by thanking the organisers for the opportunity to present some of our work here, and hopefully in the next year or two, you know, there'll be some updates coming through on how successful these T cell therapy programs are. Thank you.
Thank you, Corey. A fascinating review of very innovative work. We'll all be awaiting the results of the ATA 188 studies with great interest. Amanda, I think, has been collating questions.
Yes, we've had a couple come through, and everyone please feel free to keep asking questions as we go along. Katie has asked some good questions. Is the data showing that if you contract EBV pre-adolescence, your risk of contracting MS is much lower? Also, is there any evidence around severity of the original EBV episode linked to the likelihood of contracting MS?
That's a question for Mitch.
Yeah, typically when you contract EBV as a younger child, the first 5 years of life, you don't have a lot of symptoms. Some people may have some mild symptoms. So it's only when you hit your adolescent years and you develop a monotype syndrome. That's only about 50% of people that are infected. But the evidence is the later you're infected age-wise, the more likely you are going to get MS and get it more quickly. So I'm not sure if the association of the type of MS, the more severe MS, has been studied in great detail, but generally the later you acquire it, the more quickly you would develop MS in terms of time. I can't comment on severity based on the severity of the mono, but higher titers tend to portend more rapid onset of MS from the work that Alberto's done and others.
Great. The regional, next question was, the regional map indicates that MS incidence may not actually relate to latitude. So for example, South America incidence does not seem to be as high as Australia or the UK. Are there any other theories on why some geographic locations have a higher prevalence? Maybe a link to some time outside of vitamin, maybe a link to time outside, such as vitamin D exposure or stress levels?
Good question. How do you explain the gradients and what's going on? Some of it is just a lot of places in the world, including the Middle East, have just started doing more accurate surveillance. That's part of it. And there's just a lot of places where there's not enough neurology expertise to diagnose MS. There are things that maybe could be turning things on, including sunlight, including issues around weight, other stressors in the body. But I think there's a lot of dynamics going on right now in many places of the world where we're finding new data. And really the key thing for risk is incidence, number of new cases. I presented the map of prevalence. Prevalence is all. So that's more the burden of disease in terms of how many people in your community or country have MS. But the incidence measure, a little harder to get at, the number of new cases, is really important for risk. And so those measures are a little more challenging to get, but we see that there's more, the gradients are maybe not as steep with incidence. Nonetheless, I think the stuff going on in South America, in the Southern Hemisphere, is very interesting. The latitude gradients are reversed, and they appear to be still present. And so, we have a paper coming out looking at a new updated model for at least the United States, and shows the gradients are still there.
So can I say something? There's one area of EBV research that people have been interested in for a long time. So EBV it's a virus, but it obviously has variants and lots of regional variants. And it's never really strongly been proven with any of the EBV-associated cancers, but the suggestions that you may have certain variants of the virus that may be associated with certain disease risks. So that is something that people will start to look at in MS, whether there's a virus, a pathogenic virus that has differences in its genome that may play a role. So there's obviously the host genomes, there's the genetics, but is there some viral genetics on top of that? So, but it's by no means is it clear at all that is playing any role in MS, but that's an area of research that people are going to start being interested in. Are there types of the virus that may increase risk? Yeah. Great.
It'll be interesting to follow that. And one more question here, and then I've got a couple more on the other area. Does Epstein-Barr virus behave differently in females versus males? Just wondering why females are more affected by MS if one of the primary causes is EBV.
That one's for me, right? It's a good question. The group that in London looked at the EBV epidemiology in general, I'm trying to think of sex difference. I mean, there was females tend to have more rapid acquisition of EBV. And when they look at a population-based sample, there can be different symptoms, even of mono, in males versus females. So there probably is some acquisition and also symptom-wise differences in men versus women. I'm not sure if that's been carried out and how that would relate to differential risk for MS. I know that in the Ascherios work, both men and women seem to get MS at similar rates when they're infected with EBV in terms of those graphs I showed. I think that I have to look in more detail and even ask Alberto if there's a difference in terms of the load and antibodies and risk for developing it, and if that's any different by sex. But there are some subtle differences, looking at the epi and how it presents clinically. I'm not sure how that would weigh into questions about MS though, but something to look into.
Just on differences in sex from an immunological perspective, that there is data now coming out, not in EBV, not in MS settings, but thinking about the different impacts sex hormones have on our immune system. And it's clear that there are differences in the way we generate immune responses, and whether those things will ever play out in risk is a lot of work needs to be done. In the case of EBV immunology in general, I don't think anyone's ever looked at differences that arise, had a good look at differences that arise, say, for example, in females or males. I don't know of any studies that looked at that. Yeah.
Great. A question which was actually in my mind, which someone else has kind of put forward, is there a possibility that stem cell treatment could remove EBV from your cells, or will it always be there? Is it possible that this would stop MS progression?
Yeah.
I guess with it being such an interesting topic at the moment as well, what's the relation like?
Can I take that one? That's an interesting question, and it's something we've discussed with our colleagues here. And it's a similar question for the B cell depleting antibodies. Are you going to have and it's something we're going to start looking at. I don't think so. Because obviously EBV is a problem in people who get stem cell transplants for other reasons. And I'm not sure in those settings that the virus ever disappears. So if you've got you've got leukemia, you get a stem cell transplant, you've got virus. I think you don't ever lose the virus, but I'm not 100% sure. But certainly you would reduce the burden of virally infected cells in that setting. If you're doing a stem cell transplant on the back of depleting someone's immune system, within that immune system will be EBV infected cells. And they tend to not make up a large number. So yeah, it'll have an effect. It's something we've talked about with some of the neurologists in Brisbane, looking at, maybe not in this setting, but looking at some of the anti-B cell drugs, ocrelizumab, potentially rituximab, and looking actually what happens to EBV. The hard thing is it's not easy to find, to tell you the truth. I don't think even in MS patients it's particularly easy to find. You can't necessarily see it in circulation. You may see increased in the saliva, but in most people it'll only sporadically come out in your saliva. It's in 1 in 10,000 to 1 in 100,000 B cells in our blood. There doesn't seem to be increased in MS patients. So it's a good question. It's not an easy question to answer, to tell you the truth. And we're going to try to look at it from the angle of EBV immunity, which is an indirect approach, but also looking at whether we can find evidence of EBV reactivation, EBV infected B cells, etc. Yeah.
And that kind of question, your answer is leading on to what the next question is around testing, potential testing for EBV. Is it something that, is recommended, or you think in the future that might be an option for people to look at?
I think, I don't know, whether Mitch would agree, general testing for EBV is not going to tell a lot, because you test everyone, eventually 95% of people are EBV positive. And I don't know how clear-cut antibody titers and things are, but yeah, maybe in familial approaches, etc., you may be able to get to those stages. Potentially at early stages of the early stages, in CIS and stuff, of early diagnosis of MS and things. But yeah, I don't know if Mitch wants to add anything to that, but I'm not sure.
We don't usually, at least by the time people come in and they present with an MS symptom, they're in their 20s or 30s, most cases. It's not usually a typical test. I mean, most people, are 95% of the population is infected, and the EBV titers, even the antibody titers, can go up with other infections too. So it's right now, at least, it's not that helpful, at least in terms of pinning down a diagnosis. And there may be some other kind of markers that could be developed, but I don't usually test for it.
Those are the questions that we have, unless anyone has anything to add into the Q&A or the chat function. Ernie, is there anything that was raised for you?
I had a question that Corey might be able to address perhaps, which is a link between EBV responses and environmental factors. I think there's been some work suggesting that low vitamin D levels might affect the nature of one's response to EBV. Is that sound work, or?
I'm not sure. Only I'm not aware of that. But it would make potential, some sense. EBV immunity is so complex, like a lot of immunity to these persistent viruses. For example, there's been suggestions that you have increased, responses to the lytic antigens in MS as well, and not necessarily linked to vitamin D, but, so it's a good question. It's something we should be looking at. There's actually groups at QMR that actually, look at a lot of vitamin D. There's a lot of good epidemiologists at QMR as well, but it's I don't think it's clear. And I think the layers of complications on that are HLA genetics as well, as Mitch talked about. How do the HLA genetics and vitamin D interact with our EBV immune responses? Yeah, and it's something that really would require very large studies. And from my perspective as a T-cell immunologist, they're not easy studies to do for T-cell immunology. It's potentially, possible studies for antibody responses, and looking at the impact of vitamin D in that space. Yeah.
Over to you, Annie.
Right. Well, I thank both speakers. Very impressive, very clear and valuable talks. We appreciate both your time and your expertise, and hope to see you around at meetings in the future, perhaps. We'll catch up.